All right, guys, moving on to lecture 15 and 16. I'll probably do 15 in this one video, then I'll do 16 in the following, but we're going to be talking about amino acids and urea metabolism. So this one is probably the most complex lecture in terms of conceptually, in my opinion. There's just a lot of new pathways. It's almost like everything is separate, but not really. And I'll explain why in a little bit. But I just want to give some background before we start talking about the urea cycle, before we start talking about everything. I just want to talk about transamination reactions. So we've already seen this a couple of times with the malate aspartate shuttle, right? Where we basically transfer an amine group from our amino acid to an alpha keto acid, right here, to a keto acid. Or it's not alpha, I just said that, but to a keto acid. And so we transfer it and we flip it, and it's essentially not necessarily using up ATP or anything like that. It's just transferring the amino group or the amine group. And in order to do this, the amine group needs the PLP, pyridoxal phosphate, which there's a mechanism, but I honestly deprioritize it. I don't know if it was mentioned in your guys' lecture even. Honestly, I'm just going through this. But yeah, essentially this is the mechanism of how this whole thing happens. But it's pretty, self-explanatory. You have a base of your thing, deprotonates, causes a shift base to occur. When the shift base is here, the shift base will end up collapsing here. And then from there, it'll form this intermediate that basically pushes back, and then we see that the amine group leaves, or something like that. I don't even know, but it's a mechanism. I just deprioritize it. And yeah, and so basically the amino acid will become our keto acid. The keto acid will become our amino acid. So just things to note. So moving on to the next one, glutamine transports ammonia from tissues to the liver. So glutamine, we know that it can be transported in non-liver tissues to the liver and vice versa, and so on and so forth. And so what I wanted to talk about is that glutamate will be converted into glutamyl phosphate, which will then be converted into L-glutamine. And so what I wanted to say was, for glutamate, it's going to convert into glutamyl phosphate, which is just simply a glutamate with a phosphate group attached via this enzyme called glutamine synthetase. And it's going to add an ATP or a phosphate group from the ATP and add it to our glutamate. And then from there, we're going to add an amine group, and it's going to replace our phosphate group, and it's going to form the amide, which is now considered glutamine. So now this is glutamine synthetase. It's the same enzyme, just has a two-part thing, essentially. So for glutamine functions as a temporary nitrogen storage, where glutamate contains two nitrogens, the amide, and then also the amine. And then specifically helps transport ammonia to the liver, where it can be detoxified and excreted through the kidneys and the renal system. And so glutamate is charged and ammonia is toxic. So glutamine is polar, but at least it's not charged, so it can travel through the blood. And it's just not charged. It has extra issues with viability in the cells and whatnot. And then in the liver, glutamine gets converted to glutamate and ammonia. So as you can see here, the glutamine gets converted back to glutamate, and the ammonia is released through the urea cycle. So the enzyme that does this is called glutaminase. This is something that you should know in terms of how we get ammonia to the liver, or specifically to the kidneys or in the liver. And then from there, it gets through the urea cycle. Yeah, I think I misspoke on this slide. So now talking about glutamine. Wow, I did not mean to make myself full face. Okay. So now talking about glutamine transporters, or glutamine being a transporter for ammonia from tissues to the liver. So we know the liver is going to basically excrete this ammonia through the urea cycle. And so the way it does so is we first have glutamate that uptakes, or that basically it's going to add an addition of this ammonium group, where our glutamate is going to first be phosphorylated with ATP. And once it's phosphorylated, then we can add the amine group, ammonium group, whatever, and attach it and form glutamine now. This is an amide right here, in that we know that this is glutamine, the structure of it. And then from there, once glutamine makes its way into the liver, glutaminase is going to only be found in the liver, and it's basically going to break apart the ammonium group and take it off. And then we form glutamate finally, and then the ammonium group will be excreted through the urea cycle. So this is just a way to specifically transport ammonium to the liver, and then glutamate is charged and ammonia is toxic. So if we combine the two, we basically get the best of both worlds, if that makes sense. It's no longer charged, and then ammonia is no longer free, so it's not as toxic as we expect it to be in the body. So moving on to the next one. So alanine transports ammonium from active muscle to liver. So this is just general tissues over here. This previous one is just talking about tissues that just do this process on its own with glutamate. Now in skeletal muscle, we use alanine because alanine is the amino acid for pyruvate. Pyruvate is the keto acid of alanine. So we know that glucose will undergo glycolysis to form pyruvate. Well, what happens here is our amino acid, blah, blah, blah. So basically what ends up happening is the amine gets transferred onto alanine. And alanine can go through the blood, it can transfer, and it's going to make its way to the liver. Once it's in the liver, it's going to get rid of the amine group. The ammonium is going to do the urea cycle. It's going to get excreted and eliminated. And then we form a pyruvate. And then pyruvate goes through gluconeogenesis in the liver. Remember, that's a site of gluconeogenesis. And then it's going to form blood glucose, and then it's going to go back to the muscle. So, again, alanine transports ammonia because there's a ton of pyruvate in muscle, which converts to alanine with this alanine aminotransferase. So that's something to know, the enzyme and what converts it. And then in muscle, pyruvate plus glutamate forms then alanine and alpha-ketoglutarate when we subject it to this enzyme. And so alanine enters the bloodstream, as I explained this diagram, and that's just another way of transporting ammonia to the liver, but this is skeletal muscle. So know the distinction between general tissues, which uses glutamate, and glutamine, where this one uses pyruvate alanine. So good to know. And then glutamate donates amino groups in the liver. As we mentioned, they basically convert it to a glutamate, and then from there, it'll be converted into alpha-ketoglutarate. And again, glutamate often acts as an amino group donor in transamination reactions, converting into alpha-ketoglutarate in the process, and usually the enzyme that does this is called glutamate dehydrogenase, which is a redox reaction. We end up reducing this enzyme. We end up oxidizing glutamate and then reducing our cofactor to form NADH or NADPH, one of the two. Yes. And then there are three major excretory forms of nitrogen, right? When we have this ammonium, there are different ways we can excrete it. So there's ammonium, which, as we know, is alkaline. It's used mainly by aquatic animals, but it can be converted into urea, which is a neutral charge. And it's high water loss when excreted, and then it's in many vertebrates. And then the third one is uric acid, which is insoluble because, again, you don't really have the polar groups. You have the rings, like kind of making it non-polar. And then it excretes without a lot of water loss. And it's usually in birds and reptiles. You should know all three, but prioritize urea, and it's foreshadowing that urea cycle. So now we're going to start with entering the urea cycle, right? This is rate limiting step of the urea cycle. So before we get into all the three different loops, we're going to talk about, the entry point into the three loops. So think of this I'm just going to leave it like that. And so we need to form carbamyl phosphate to initiate the urea cycle. So overall goal is to dispose and excrete nitrogen. And nitrogen enters the urea cycle via carbamyl phosphate. So as you can see here, we start with bicarbonate, we have ATP, we use biotin or all that stuff or whatever, and we basically convert it to this carbonic phosphoric acid anhydride. And then from there, when basically all it is like the bicarbonate and the ATP get attached, and then the ammonium gets added into the mix, and that forms our carbamate. Then once we have our carbamate, we use another ATP as well to form our carbamyl phosphate. So again, this is the three-step process. I have it written in words, essentially. You don't really need to know this complex word, just know intermediate one. Carbamate you should know. But essentially, we use 2 ATP to start this initial process, right? So just remember that too. And this is the rate-limiting step of the urea cycle, and it occurs inside the mitochondrial matrix as well. You have to know spatially where these locations are. So when I know I mentioned in a couple of videos before that you should draw all the pathways and specifically in their different compartments. Make sure you write them in their respective compartments. And even though there may be similar enzymes, if they're in different compartments, that changes the game a little bit because molecules can't just freely diffuse. And we know this. And I just want to emphasize the enzyme that does this entire process is called carbamyl phosphate synthase 1, CPS1. So now we enter the urea cycle. This is just one of the loops out of the 3 total. So for this one, we see that carbamyl phosphate's on the top, and it's going to combine with ornithine, which is not necessarily an essential amino acid, because essential amino acids just mean that our body doesn't produce it and we have to take it in from dietary intake. But ornithine is a non-essential amino acid, which is essentially our body produces it and it's on its own, so we don't really need to consume it. So these two combine and we form citrulline, right? The blue is the amine that's going to get excreted, right? We're going to get rid of it. So the citrulline, we have the carbon here. The yellow one is I'm pretty sure it's from the urea. Yeah. turn this TV off, turn this TV. Yeah. No, it's not from the urea. This carbon, I think it's this up. This one right here from the carbamyl phosphate. So citrulline gets added, and then from here, the citrulline is going to go from the mitochondrial matrix to the cytosol through a transporter. You don't really need to know the specifics, but we're going to add another ATP, and this is going to form our citrulyl AMP intermediate. Just know it's a citrulline ATP or AMP intermediate. And then from there, what's going to end up happening is this will power the second part of the reaction, where we're going to add aspartate, and we're going to replace the AMP with the aspartate, right? So as you can see here, we have our citrulline on the right, and then we have our aspartate coming in right here on the left, and this is forming argininosuccinate. And basically from there, when we form this, we're going to cleave it into two molecules: fumarate and a plerotic reaction, or into arginine, which again has this ammonium that we want to excrete, right? Same with the aspartate, it also carries that ammonium that we're going to excrete. For step number 4, we're going to hydrolyze the urea off of this arginine group, and we're going to form ornithine. Ornithine is it's like a derivative of our arginine, essentially. And so the urea gets hydrolyzed off, and the ornithine basically goes back into the mitochondrial matrix through a shuttle system, and again, proceeds the cycle over and over and over. And again, remember that this uses 2 ATP equivalents. One right here, even though it's 1 right here, it's technically 2 because the 2 pyrophosphates go off, and we have an AMP attached to it, not an ADP. Hopefully that makes sense. So now supplying aspartate for the urea cycle, we know that we need aspartate, so where does it come from? This is what I was talking about. It's that like complex 3 loop. Remember that malate aspartate shuttle? Remember how like before I was like, oh, you don't really need to know the aspartate, just know that it's there? Well, actually you do need to know it. It's a part of the urea cycle. So that portion that we talked about, that aspartate, is actually going to be used for the urea cycle. So so the premise of the urea cycle and how we regenerate it is that we need aspartate as an input, and the citric acid cycle produces oxaloacetate, right? And the oxaloacetate is a precursor to aspartate. It's the keto acid of aspartate. So we use an amino transferase. What do we use? We use glutamate, and the glutamate is going to have that ammonia group. It's going to attach it to the oxaloacetate right here, right? So the oxaloacetate grabs the ammonium from the glutamate and then forms aspartate. The aspartate will then transport out via the malate aspartate shuttle, and from the aspartate, we're going to combine it with citrulline to form argininosuccinate. And so again, this is called the aspartate argininosuccinate shunt, which ultimately regenerates aspartate from fumarate. Fumarate is formed in the urea cycle after splitting argininosuccinate. We know this; it's split off here, but it's not shown. And yeah, recall that malate aspartate shuttle was used to transport cytosolic NADH to the matrix, but it also transports the ASP into the cytosol, which can be used by the urea cycle. And then it also feeds into the citric acid cycle as well. And then side note, 2 important reactions contributing to the urea cycle occur in the mitomatrix, not the cytosol. Ammonium plus bicarb to form a carbamyl phosphate. So this entry into urea cycle occurs in the mitochondria, and the carbamyl phosphate plus the ornithine, right? Combining these two form the citrulline. This is going to happen in the mitochondrial matrix. It is important and imperative that you know where these locations are, because you will be asked on the exam, like, oh, which part of the urea cycle is going to be in the mitochondrial matrix? It could be a simple multiple choice question, but that is like for sure you guys will be tested on that. So know exactly where everything is spatially. So in terms of like net cost, right? So we know the carbamyl phosphate synthetase uses 2 ATP, the first one here, and then the other one where like my head is near. This is the 2 ATPs. And then argininosuccinate formation uses 2 ATPs, 2 ATP equivalents. 1 ATP gets formed into an AMP, right? So the AMP is right here. And then we initially have the ATP, but it gets converted to a pyrophosphate, or the pyrophosphates leave from it. So we essentially use another 2 equivalents. But the urea cycle doesn't produce ATP, but produces fumarate, which feeds into the citric acid cycle. And we know that fumarate to malate to oxaloacetate, the malate to oxaloacetate step produces NADH. And NADH does what? Produces 2.5 ATP. So 2 plus 2 is 4. 4 minus 2.5 is 1.5. So that is the ultimate net cost of the urea cycle, is 1.5 ATP. Remember, we form our fumarate, which is on, and I'll just reiterate, it's right here on this bottom left part. So now we're going to regulate the rate limiting step of the urea cycle, that carbamyl phosphate formation. So the way we do so is CPS1, carbamyl phosphate synthetase 1, can only like really be stimulated, activated through N-acetylglutamate. So N-acetylglutamate is just a glutamate with a acetyl group attached to the nitrogen on the protein backbone. So as you can see here, this is our alpha carbon. This is the amine group or the amino group, but it's not really, because we see that we have an acetyl group attached to it instead of another H. you know? So this N-acetyl directly stimulates and activates this. That's the direct regulation. Now the indirect is that the arginine from the urea cycle, if the urea cycle has all the intermediates ready to go, the arginine is going to stimulate this N-acetylglutamate synthase, which is just going to generate this product. So if it stimulates this product generation, then this is going to end up ultimately stimulating. So it's indirect is all I'm trying to say for this part right here. The arginine will indirectly stimulate this production, and then the production of this product will stimulate our enzyme that is the rate limiting step. Yeah. And then here's an overview of the amino acid catabolism now. So moving into like how amino acids could be ketogenic or glucogenic, or they could be either or. We'll go into specifics. So ketogenic amino acids just means that we can turn these amino acids into ketone bodies. And the glucogenic amino acids just mean that they can be broken down from amino acids into glucose molecules or into TCA intermediates. And the only observation I will say is that leucine and lysine are the only exclusive ketogenic amino acids. So a lot of these ketogenics can actually be gluconeogenic as well, but leucine and lysine are the only ones that are not at all. So just want to emphasize first, read this note on the side. The color coding is reversed from the previous slide diagram, so don't let that trip you up. Here, the ketogenic is orange, but on this other slide, the ketogenic is actually blue. So I just really wanted to emphasize that, don't be tripped up by the colors. But this is what I wanted to say. Lysine and leucine are the only true ketogenic, whereas tryptophan, threonine, phenylalanine, tyrosine, isoleucine are ketogenic, but they're also gluconeogenic. So they could be either or. It just depends on how they feed into the pathway, which enzyme interacts with tyrosine first. And then all of these molecules on the right are exclusively gluconeogenic. You will not find these as a ketogenic amino acid. And then there are 5 important cofactors for amino acid catabolism. And actually, before I move on, I just want to emphasize as well on this previous slide, you do have to know kind of which amino acids feed into what part of the TCA cycle. It is important to know because they may ask you and I have a practice problem on this, okay, what's the amino acid that feeds directly into alpha ketoglutarate in one step? Well, that's glutamate, and so you would have to know these. So definitely know these pretty much by heart, but I think the more important thing is that you know which one is ketogenic and gluconeogenic, and then worry about this other stuff about where it feeds into later on. I mean, it should make sense though, because if it's glucogenic, we know that it can feed into an anaplerotic reaction, right? If it's glucogenic. If it's ketogenic, it can't go as an anaplerotic reaction, so it ends up forming acetyl-CoA. So kind of like a little tip in terms of oversight or overview. So 5 important cofactors for amino acid catabolism. There's PLP, which we know is an essential cofactor for transamination reactions, shifting that amine group. We know biotin does the carboxylation reactions, and it uses ATP. We know adamant does a 1 carbon transfer using a methyl, not a carbon dioxide. And then we know THF is a 1 carbon transfer, which is an intermediate oxidation state. And essentially, it's just an aldehyde functional group that it attaches it to. And then THB is a redox reaction. And so the reason why I'm bringing these up is because you should know the structures of them, because you may be asked to recognize what type of cofactor coenzyme is this. And you just have to recognize and write down the name of it. So going on to the catabolic pathways for tryptophan, alanine, serine, cysteine, and threonine, glycine, so the gluconeogenic stuff, all of these are ultimately going to be converted into pyruvate, because we know pyruvate can be converted into glucose via gluconeogenesis. That's the whole point. It's it'll feed into the Krebs cycle, but ultimately it'll become pyruvate, and then from pyruvate it goes upwards. So specifically, cysteine is going to become pyruvate directly. Tryptophan is going to be converted into alanine. From then, alanine will be converted to pyruvate, and then threonine plus glycine will combine to form our serine. The serine then gets converted into pyruvate. So I was just looking at this bottom left. So here's the mechanism of a lot of these. I would say deprioritize all the specific enzymes, but definitely you will have to I would recommend memorizing these as well. This exam, I will preface right now, it is probably the most memorization heavy exam out of all of them, but this is the final time you guys will memorize so much information in such little time. Exam 4 should be a lot more bearable in terms of what you're going to memorize, all that stuff. So this is going to be the last final push of memorization, putting everything together. So we have here threonine dehydrogenase. We have 2-amino-3. Yeah, these are the enzymes specifically that will convert the threonine. And we know that the threonine is going to be added with the glycine, which the glycine is here. And then here's some diagrams of like how everything is integrated together. So like the tryptophan right here is gonna be converted through 4 steps into alanine, and then from alanine gets converted into pyruvate. And then from here we have the threonine plus the glycine, and it's basically gonna form our serine right here. And then from the serine, we're going to convert it into pyruvate from there. And then we know cysteine through 2 steps will become pyruvate. So like this, you just need to know specifically how they feed into pyruvate and like how many steps or which amino acids are used. And that's pretty much it. And then these are the catabolic pathways for the phenyl and tyrosine, which are both ketogenic and gluconeogenic. So ultimately, phenylalanine and tyrosine are going to be converted into acetyl-CoA and fumarate. We know fumarate can be an anaplerotic reaction for the Krebs cycle, and then the former, I'm not going to say the name again, but can be used for ketone bodies to be broken down to 2 acetyl-CoAs. And we know acetyl-CoA is not an anaplerotic reaction. It can't fulfill any of the intermediates in invertebrates because we don't have the glyoxylate cycle. So phenylalanine will produce pyruvate in 7 steps. Do you have to memorize everything? No. No. You do not have to memorize this conversion all the way to fumarate. The second thing, tyrosine will get converted into acetyl-CoA in 6 steps, right? And it's right here, but I don't think I really showed tyrosine here at all. Or technically, yes. But it'll be converted in 6 steps all the way to acetoacetate, and we have our fumarate here. So it's basically like both these are combined, but ultimately they get cleaved on the last step. And then from there, the acetoacetate will become acetyl-CoA in the last step, and by this enzyme itself. And so enzyme defects lead to conditions such as like PKU, tyrosinemia, alkaptonuria, which honestly I don't even know, but you would have to look those up if you wanted to. But these are just like extra little minor details about it. I don't know if it was really emphasized as much. Okay, so moving on to the branched chain enzymes. This one's a little bit easier to understand. So basically these are both glucogenic and ketogenic enzymes. So this is where we have leucine, isoleucine, and the isoleucine valine. So these are the branched amino acids, right? So valine, we can see it's branched here. We know isoleucine is also branched as well, and then leucine obviously is branched. So we're going to undergo this enzyme, which is the branched chain amino transferase. So it makes sense. It's like a general enzyme for the branched chain amino acids. It'll take off the ammonia groups from all of them, essentially, and form alpha keto acids respectively to each one. And then from there, it's going to use this branched chain alpha keto acid dehydrogenase complex. And a lot of these cofactors you've seen before in PDH, pyruvate dehydrogenase complex, CoA-SH, TPP, FAD, lipoate, NAD, CO2. It's a very similar enzyme, but instead of using pyruvate, it's going to use these branch chain alpha keto acids, not amino acids, keto acids. And then from there, we form our activated intermediates, acyl-CoA derivatives. And these are going to form succinyl-CoA or acetyl-CoA or acetyl-acetyl-CoA and then get broken down to acetyl-CoA. That's all pretty much you really have to know. And then this branch chain dehydrogenase complex can be inhibited by maple syrup and urine disease. No idea. Basically, it's this type of drug called sotalone, which basically inhibits this, and then therefore you're stuck and you can't break these amino acids down. Maple syrup urine disease, I think, or something like that. I don't really know. Catabolic pathways for aspartate and asparagine. We know that these are glucogenic, and they're ultimately going to form oxaloacetate. We know specifically asparagine gets converted to aspartate, and then aspartate, we know from the aspartate, the malate aspartate shuttle, it gets converted into oxaloacetate. So as you can see here, asparagine will basically have this amide group be converted into a carboxyl group by asparaginase, and it forms aspartate. And then from aspartate, we use aspartate aminotransferase, PLP, as a coenzyme, and it'll basically get converted into oxaloacetate. And we use alpha-ketoglutarate as our other like substrate that will accept the ammonium group as well. And so here's a practice question, like kind of like helping you understand what specific enzymes are going to be feeding into where exactly. So yeah, hopefully that was a little bit helpful. So I'm just going to go through this. Those are basically the answers. And then moving on to amino acid biosynthesis. So biosynthesis uses glutamate and glutamine. So we know glutamate synthetase, which is this diagram on the left, you've seen it before, is basically converting our glutamate into glutamine using a 2-step process of a single enzyme, and it's going to add a phosphate group and then eventually add the ammonium because now it's like activated. And then for glutamine aminotransferase, this is a different one that you guys really haven't seen as much. But essentially, we have our glutamine, right? And what ends up happening is this enzyme is going to have a cysteine attached to it. It's going to attack that amide carbon. And then from there, it's going to basically cause this ammonium to leave. And the ammonium has actually a channel it can go through and basically bind onto the active substrate and get converted into urea, essentially. That's like the end goal. And then our glutamate is able to leave as well in this reaction when it's just, when water is introduced. That's pretty much like the two different types of amino acid biosynthesis using these two different pathways. Okay, and then moving on to regulating amino acid biosynthesis through glutamine synthetase, and there's two ways to do so. So there are 8 total allosteric regulators, and I know that's a lot. I know there's more to memorize. I'm so sorry, guys. But so basically, these are essentially all feedback inhibitors: histidine, tryptophan, carbamyl phosphate, glucosamine 6-phosphate. All of these are going to negatively basically inhibit glutamine synthetase. So it's going to stop glutamate from being converted into glutamine and to capture that nitrogen, that urea cycle, essentially, the amino acid biosynthesis. So these are metabolic status inhibitors. So like CTP, AMP, glycine, alanine are also going to inhibit it, but they're more so like about like energy levels. Glycine and alanine are general indicators of amino acid metabolism in cells. Okay. So now talking about other ways of regulating is covalent modification. So adenylate a tyrosine residue on glutamine synthetase. So glutamine synthetase, again, same this enzyme, but another way we can inhibit it is we adenylate it with at this specific residue. So tyrosine 397 is a specific residue on the glutamine synthetase, and if we adenylate it, meaning we attach an AMP onto it, we can actually inhibit it. So as you can see here, adenylyl transferase is another enzyme that interacts with this and basically forces this AMP to be bound to the tyrosine, and it inactivates it, so it's more sensitive to inhibitors now with this inactive glutamate synthetase, right? And then another thing, there are additional layers of regulation, which makes this much more complex, but let's break this down. So there are additional layers of regulation. So adenylyl transferase that does this process. So remember, adenylyl transferase is an inhibitor, right? Well, we can also inhibit the inhibitor, right? This uridyl transferase will inhibit adenylyl transferase. And if this is no longer going, if this arrow is X'd out, then we know that the active glutamine synthetase is now less sensitive to inhibitors because it's not adenylated. Hopefully that made sense. I will repeat it one last time. Uridyl transferase is going to inhibit this adenylyl transferase enzyme. So this arrow is no longer like, let's just, that right, this, all the stuff to the right is gone. X'd out. So now what we have as a majority is the active glutamine synthetase. And at this state, it's less sensitive to inhibitors and it's more active. So in a way, uridyl transferase is an activator of glutamine synthetase. Just another way to put it, it's an inhibitor of an inhibitor, if that makes sense. And then here's another layer of it, just to get more complex, is uridyl transferase will also be regulated as well. It'll be regulated by glutamine, as in it'll inhibit. So glutamine, remember, is the products, right? If we have a lot of the products, we don't want to generate more. So glutamine inhibits uridyl transferase. This arrow is gone, and adenylyl transferase is at 100% functionality. So that means that the active glutamine synthetase is going and operating at 100% capacity here. And so we now are, and since it's operating at 100% capacity, we see that it is going to be more sensitive to inhibitors, and glutamate synthetase is going to have less of a function. So this makes sense though, because glutamine, we already have a lot of product. We don't want to generate more, so we want to stop this from going. So that's the mechanism of action, but the end result is we stop producing glutamine from the glutamate. Hopefully that makes sense. And then we also know that alpha ketoglutarate is is an activator for uridyl transferase. And if it activates this, it's going to inhibit adenylyl transferase, and it will keep the glutamate synthetase active. And that's because alpha ketoglutarate is a keto acid for aspartate, right? And we know aspartate is going to be used in this pathway for biosynthesis. If we have a lot of keto acid present, then we want to convert it into amino acids. And this is what amino acid biosynthesis is used for. Or this is what this pathway is used for, is for amino acid biosynthesis. So that's another thing. So the net result is that glutamine synthetase is more active at high alpha ketoglutarate, high ATP, and low glutamine, low inorganic phosphate. And then it's less active inversely. Like if we have low alpha ketoglutarate, low ATP, high glutamine, high inorganic phosphate. So overview of the amino acid biosynthesis. So there are 6 total groupings of amino acid biosynthesis based on 7 precursor molecules: pyruvate, oxaloacetate, alpha ketoglutarate 3-PG, erythrose 4-P, NPEP, and then ribose 5-phosphate. So if I were to prioritize one, prioritize the 3-PG. Prioritize this family because you actually cover this one in depth, which is, we know that it gets converted into serine from erythrose 4-phosphate, and it's reversible. And we know that erythrose 4-phosphate can also be converted to tryptophan, phenylalanine, tyrosine to form our aromatics. But just know that this is like the overview of the amino acid biosynthesis as well. And then the precursor molecules from glycolysis, citric acid cycle, and the pentyl phosphate pathway produce amino acids that tend to share similar structural properties. So, before I move on, one thing I wanted to emphasize is that for the reason why high alpha ketoglutarate stimulates this and makes the glutamine synthetase more active is because in this next slide, you can see here that in the citrate acid cycle, that's what this loop is, the alpha ketoglutarate gets broken down or converted into glutamine. So this is our reactants, and this is our product. So if we have a lot of reactants, we obviously want to stimulate the generation of products. So that's exactly why in this one, it ends up stimulating. So just as to how this relates to amino acid biosynthesis. So now moving on to the next part. So a closer look at the 3-PG family. So ultimately, we have 3-PG gets converted into serine, and then it gets converted into glycine and cysteine. And essentially, the glutamate donates amino groups, and the THF enables 1-carbon transfer. So you can see here on this left diagram, we have 3-PG. It's going to get oxidized, and we're going to form our 3-phosphohydroxy And so as you can see here on the starting molecule versus this one, we see that we have a hydroxy group right here, right? And it turns into a ketone group right here. And then from here, what's going to end up happening is we undergo a transamination reaction, and we form our 3-phosphoserine. So we're going to get rid of this carbonyl, and we're going to replace it with an amine group, with the phosphoserine aminotransferase. And then from there, we're going to lose the phosphate group using a phosphatase enzyme, and we form our serine. And then our serine hydroxymethyltransferase gets converted and will convert our CH2OH, or basically it'll convert our serine into glycine by removing the CH2OH from it. And so that's ultimately the fate, is that it'll either form with glycine or cysteine. And so I just want to talk about here, or the serine can combine with homocysteine to form our cystathionine, and from there it'll break apart and form our cysteine. So basically serine can go 2 pathways. It can go the glycine pathway or the cysteine pathway. And I just want to talk about that the S in cysteine comes from methionine, an essential amino acid. Yeah, and then superficial notes on some of the other families. The amino acids derive from oxaloacetate and pyruvate, and here. Use ATP and NADPH, receive amino groups from glutamate, and then they use THF as a cofactor for the 1-carbon transfer, the methyl groups. And then aromatic amino acids share common precursors, right? So right here, the aromatic amino acids, they share as PEP and erythrose 4-phosphate. These are the common precursors, and the intermediate is chorismate, which is formations inhibited by glyphosate, which is, again, you don't really need to know this stuff. This is just more superficial things that you should definitely prioritize last. Phenylalanine and tryptophan are essential amino acids, as we know. Vertebrates can't synthesize this on their own. And then lastly is the ribose 5-phosphate exclusively makes histidine. And it's because it makes PRPP, or the ribose 5-phosphate gets converted into PRPP. And when it gets converted into PRPP, basically it's going to get added, ATP is going to get added to it, glutamate, and then it'll form histidine. But the most important thing out of this 4 substrate reaction is that PRPP is the most important part of it. Yeah. And then example of amino acid biosynthesis regulation in the oxaloacetate family. So if we're talking about specifically right here, where we have the aspartate, asparagine, methionine, threonine, lysine, this is where we have all this. It's just broken down more in depth. So it's primarily through feedback inhibition. So if we have a product, it's going to negatively inhibit backwards, right? And this makes sense. If you have a lot of products, why would you want to make more? And then we also have enzyme multiplicity. This is a new concept where we have more than one enzyme that can catalyze a reaction. So for example, on this top part, in order to convert aspartate to aspartyl beta phosphate, we have three different types of enzymes, whether they're isoforms or just different enzymes in general. They can do this, and these different isoforms of enzymes can be regulated differently, right? A3 is inhibited by threonine, but not inhibited by lysine or isolysine, right? Because isolysine and lysine, these ones go back and they inhibit only A1, not A3. So that's what enzyme multiplicity allows us to do. And so one variant is always unregulated, which is the A2, and that maintains like almost the basal function, right? And then removing inhibitors on the other enzymes will just essentially speed it up now that we have different pathways, more enzyme available to do this conversion in the first place. So here's some more practice problems. Like, definitely go through this, memorize. This is the part where you just have to memorize a lot of it. It's not intuitive. But now moving on to a little bit of the more intuitive stuff is the nitrogen cycle. So there are three parts to the nitrogen cycle. There's nitrification, there's denitrification, and there's nitrogen fixation. And the best way I can show this is not on this slide. It'll be on the next one. But it's through this like circle thing. So we go through the first one, which is nitrogen fixation. And essentially what we do is we take nitrogen from the atmosphere and we convert it into ammonia. That's nitrogen fixation. Then nitrification is we take ammonia and we convert it into nitrite. That's what nitrification is. And then nitrite can be converted into nitrate, and that's still part of nitrification. Or nitrite can be converted back into atmospheric N2, which is called denitrification. So just going back on the slide now that we have that sorted, we know nitrification is the oxidation, or we oxidize NH3 to NO3, to nitrate or nitrite, one of the two. And then denitrification is we take the nitrite or nitrate and we convert it to atmospheric N2. And then nitrogen fixation is we turn atmospheric nitrogen into ammonia. And that's basically it. And then you do have to know that we use 6 electrons to reduce it to convert it into ammonia to do so. And then the nitrogen can be incorporated into amino acids and other reduced nitrogen carbon compounds. And 2 nitrogen fixation enzymes. So nitrogen fixation is converting atmospheric nitrogen into ammonia. There are 2 enzymes is dinitrogenase reductase, which transfers electrons one by one from ferredoxin to dinitrogenase. And it requires 2 ATPs per electron. So if it does 6 in total or something like that, then it's going to cause 12. Something like that, I think. But dinitrogenase is the second enzyme where essentially we transfer electrons from dinitrogenase reductase, that first enzyme, to reduce N2 to NH3. So these work in tandem, right? They work in tandem to basically convert our nitrogen, atmospheric nitrogen, into ammonia, and it's powered through the first enzyme, which is dinitrogenase reductase. I keep saying nitrogen so much. And I think this is just generally questions. I mean, you can go through this as well through the slides. They're pretty detailed. And then here are connecting these enzymes, tracing the forward flow of electrons, right? So all I'm saying is like this is where the electrons are going. So we're transferring electrons from pyruvate to ferredoxin, right? So as you can see here, pyruvate's up here. We transferring electrons to ferredoxin. Then we reduce the first enzyme with the 8 electrons, the dinitrogenase reductase. And then once it's reduced, we add the ATP, and it's 2 ATP, so it's 16, as I mentioned before. And then from the 16, we're going to transfer those electrons and reduce dinitrogenase. And now that the dinitrogenase is reduced, we take 6 of those electrons and we convert the atmospheric nitrogen into ammonia. And that's essentially it. And I'll adjust the slide so it actually shows what I was saying. But basically, I just followed the red arrows through. And here's a practice question on it. And moving on to lecture 16 now.